The overall goal of this somatosensory event-related potential recording using orofacial skin stretch stimulation is to investigate cortical processing associated with orofacial somatosensory inputs in speech production and perception. This is done by selectively and precisely deforming facial skin in a wide range of speech behaviors.
This method can help answer key questions in speech science fields, such as the potential role of the somatosensory system in speech perception, the relationship between speech perception and production, and the underlying cortical representations. The main advantage of this technique is that event-related cortical potential associated with facial skin deformation provides a direct measure of somatosensory cortical involvement in speech processing with relatively high temporal accuracy and non-invasive brain imaging technique. Demonstrating the EEG setup will be Eva Montas, a research assistant in my laboratory.
Begin by measuring head size to determine the appropriate electroencephalography, or EEG, cap. Identify the location of the vertex, CZ, by finding the midpoint between nasion and inion with a measuring tape. Place the EEG cap on the head using the predetermined vertex CZ as a reference location. Align the cap on the head based on the modified 10-20 system. Next, apply electrode gel in the electrode holders using a disposable syringe with a dull tip. Attach the EEG electrodes into the electrode holders by matching the labels of each electrode to its specific electrode holder on the cap. Then, clean the skin.